It is my pleasure and honor to introduce Evandro Fei Fang, who actually worked at the NIH with Morton for a number of years, for 5 years. And please do follow him on Google Scholar. Some of the really cool publications, including some beautiful trends papers that I myself read, really informative. So Evandro did his PhD in biochemistry at the Chinese University in Hong Kong and CUHK, so which was right nearby where I am. And then spent a number of years, 5 years at the NIH. And now he is a group leader in anti-aging research at the University of Oslo. Again, not aging research, but anti-aging research. That's what we need to use more, I think. Congratulations on that title. And he's going to be talking today about the molecular mechanism of mitophagy and NAD+ in the brain, aging and healthy longevity. Please do sign up for Slack and ask questions. The floor is yours.
Thank you so much, Alex and Morton, for this great opportunity. I would like to share my screen now. Can you see my screen?
Yes, perfectly well.
Good.
Beautiful slides.
Thank you. So today, in the coming 20 minutes, I would like to give you a couple of stories we are doing now, including the background of NAD and mitophagy, as well as the NAD mitophagy in 3 of the projects we have been doing, including in premature aging and normal aging, in Alzheimer's disease, and AI-based drug development. And I would like also to take this opportunity to share with you the no age network we are doing now in Norway. And finally, I will give you a summary. If you check out the portraits of Picasso, from 15, 25 to 90 years, you'll find he was unhappy at the quality of life when he was 90 years old. He had wrinkles, loss of the hair and the hearing. So my group is very interested in the molecular drivers to make the wrinkles, to make him getting old. Only when we know the mechanisms, we can develop a way to cure the disease. To this angle, my group has developed 3 research topics. The first one is NAD+, as Professor Eric Verdin talked yesterday, how NAD is reduced in aging and why NAD supplementation was able to improve the quality of life. The second topic is age-predisposed disease with a focus on Alzheimer's disease. And we are interested in a cellular self-clearance of damaged mitochondria termed mitophagy, how turning up mitophagy could able to eliminate the memory loss in Alzheimer's disease. And we are actively involved in drug development using artificial intelligence. As Eric Verdin shows you the importance of NAD because it reduced with age. And when we supplement NAD precursors, it improves lifespan and health span in animals. So to more of this molecule, I would like to share with you how it produced in our human body. There are 3 pathways to generate NAD, including the kinase pathway. Cells use tryptophan to generate NAD, which is very inefficient and energy consuming. The second pathway, cells use nucleic acid to generate NAD. And the last one, which is most efficient, which is the salvage pathway, started from the nicotinamide riboside termed NR, which can go inside to the cytoplasm and can be converted to nicotinamide mononucleotide, which finally turns into NAD+. So there is a very complicated thing there. Due to time concern, I want to give you a summary. The linkage of NAD to healthy longevity. It is a coenzyme for the PARPs, which involved in DNA repair and genomic integrity. NAD is also the coenzyme for the famous sirtuin family proteins involved in metabolism, neuroprotection, among others. It's also used by CD38, CD157, among the others. So NAD being degraded by those proteins to nicotinamide, as you can see from here. Nicotinamide, if we add a riboside here, that is a precursor of NR here. And if we have phosphate of NR, it turns out to be NMN. My group has been actively involved in the contribution of NR and NMN in lifespan and health span in animals. If you see the picture of the elderly Picasso, so what are the molecular mechanisms underneath? Some very good papers have summarized different hallmarks of aging as summarized here, such as DNA damage, as Björn Schumacher mentioned just now, telomere attrition, mitochondrial dysfunction, among the others. So in addition to those 9 hallmarks of aging with Wilhelm Bohr, Mark Madison, and others, we propose that compromised autophagy is the 10th hallmark of aging. Although you may argue there are linkages or overlap between those two. However, all those hallmarks of aging, they are intertwined, linked to each other. The cool thing is NAD reduction has been linked to all 10 hallmarks of aging. That may explain why it is so fundamental. My group is interested in how mitochondrial dysfunction, compromised autophagy, especially a subtype, compromised mitophagy, and the DNA damage links to NAD reduction and contributes to aging. So what is mitophagy? If you see the video here, the green ones are healthy mitochondria. These red ones, they are mitophagy, because the pH turning out from 7 to 4 to degrade them. If we give the drug like FCCP treatment, which is a mitochondrial toxicant, you see dramatic induction of mitophagy to eliminate the damaged mitochondria. So it's a self-protective response, the mitophagy pathway. In 2014, with Morton and in Wilhelm Bohr's group, we first time links NAD as an inducer of mitophagy. Speaking of molecular mechanisms, there are many pathways. At least the NAD sirtuin pathway can, on the one hand, increase the genes as well as proteins involved in this pathway, also including the deacetylation of FOXO1, FOX3, which regulates many genes involved in autophagy and mitophagy. So there are many pathways involved here. Due to the time concern, I won't go to details, but just want to highlight my group is interested in the NAD mitophagy axis in healthy longevity. So how we do our work, we are not working directly with the humans. We use different animal models which are available in the lab. Especially my group use this cross-species platform, including the roundworm C. elegans, Drosophila, mice, stem cells, and tissues from postmortem brain, human brain. I want to start from the project number 1, majorly a summary of the postdoc work. So in addition to normal aging, there's a group of disease called premature aging. As you can see the picture, so those people with mutation of genes, which makes them grow older at the age, which looks normal for the healthy people. The disease like ataxia telangiectasia, Werner syndrome, among the others. So I want to give you a piece of data for each of the 2 diseases. For example, the mice with a mutation knocked out of the ATM has a short lifespan of 3 to 5 months, compares with a healthy one with 2.5 to 3 years. And we found that the ATM mice had very low levels of NAD compared with a healthy one. When we're giving NR treatment in the drinking water from 1 month of age, after weaning status, we can dramatically extend the lifespan to over 10 months. How about the Y type, since most of us are healthy people? And very excitingly, the NR augmentation was able to extend lifespan 5 to 10 times, or 5 to 10%. The data were in line with a similar report published by Johan Owings in Science in 2016. The cool thing is the NAD supplementation not only extends the lifespan, but also improve the health span. For example, the AT mice had a low score in spontaneous alteration performance using the Y maze. However, giving NR and NMN treatment, we can dramatically reinstall the impaired SPA to that compatible of wild type. Then we wondered whether the NAD-dependent improvement of lifespan is only this disease-dependent, or it can be applied for many premature aging disorders. Then we turned to another disease called the Werner syndrome. This, we did the work in C. elegans and in cell culture to show the Werner had a short lifespan, and NAD augmentation was able to improve the lifespan. We want to confirm this benefit is cross-species dependent. To this end, we have been collaborated with Henry Jasper at Buck Institute, with Martin, who performed some experiments, and Tor Eric Rooston. Indeed, knockdown of the Werner gene dramatically reduced the lifespan from 51 to 44 days. However, with the NAD augmentation NMN treatment, it extended the lifespan of the mutant from 44 to 56 days, even higher than wild type. And the wild type also had extension from 51 to close to 60 days. So based on those many of the work working with Morton and Wilhelm Ball, we then propose a new molecular mechanism of aging. We want to have a more unified method. We highlight the importance of nuclear DNA damage, mitochondrial homeostasis, and the communication of subcellular organs. For example, we propose when we are young, we have endogenous DNA damage, which can be efficiently repaired by different DNA repair pathways, with some of them involve the NAD-dependent PAP1 activation. On the other hand, the NAD-dependent sirtuins can also increase DNA repair capacity. For our ATM paper, we also show in the ATM deficient background, NAD sirtuin was able to improve NHEJ pathway. On the other hand, the NAD sirtuin was also able to increase mitochondrial homeostasis through three pathways, including increased mitochondrial biogenesis, eliminate damaged mitochondria via mitophagy, as well as increase the cellular resistance to exogenous and endogenous stress. Finally, may contribute to neuronal and cell survival. If you look over this animation carefully, you can see when we grow older or in the condition of premature aging disease, we consistently saw a dramatic activation of the pilation, which drives the reduction of NAD, which may contribute to neuronal death and the aging process. How we can see this is a culprit rather than a bystander, because when we pharmacologically augment NAD, we were able to increase nuclear DNA repair, mitochondrial quality, which may lead to the evidenced improvement in lifespan and health span in both premature aging and normal animal models. And there are many other pathways. So because with those premature aging disease are very well in the general population, we wondered the mechanism we noticed, like reduced NAD, defective mitophagy, are also conservative in normal aging, as well as normal age predisposed disease like Alzheimer's disease. To this end, I'm shifting my gear to the second project, Alzheimer's disease. Why AD is so important? Because there have been over 50 million people worldwide affected with AD and no cure available. By 2019, there have been over 250 drug candidates in clinical trials targeting on tau and A beta, and all failed. To this end, we thought there must be other molecular pathways are also important for the initiation of Alzheimer's disease. We checked the brain tissues from healthy people, like the hippocampus region, which is important for memory. And we see beautiful healthy mitochondria there. However, in Alzheimer's disease patients at the same age and sex matched, we continually saw accumulation of damaged mitochondria as evidenced by this halo structure. So we argue the accumulation of damaged mitochondria may play a role. To this purpose, we propose that in addition to the disease-defining pathological features of tau and A beta in AD, that accumulation of damaged mitochondria can contribute to or even exacerbate the disease progression. If this is the case, then we can pharmacologically, genetically reinstall impaired mitophagy to eliminate damaged mitochondria as a way to inhibit disease, which may provide new drug targets for the staff. Due to the time concern, I do not have time to show the data from C. elegans from stem cells, but to show a piece of exciting data from the mice, the memory experiment. So how we do the memory experiments in the mice using this Morris water maze. The theory is the mouse, it does not like to swim. The first time they take time to find the platform. However, if they can remember, they can directly find the targets. So through this theory, Eugene Ho did some experiments that using the AD mice, which is APPPS1, and this mouse had been trained for 6 times and it still does not know where the platform is. It does not have any direction and moves very slow. However, the same background of the mice, but treated with mitophagy inducer, we saw it runs much faster, swimming faster, and knows the direction. So we had more than 10 mice using an unbiased way, and we saw indeed mitophagy induction was able to improve the memory. Based on our results in human brain tissues, stem cells, 2 mouse models and 2 C. elegans models, we propose that aging, genetic and non-genetic factors contribute to defective mitophagy, which leads to accumulation of damaged mitochondria. There is a very complicated intertwined network interactions among those 4 parameters, and I didn't have time to go to details, but anyways, they contribute to neuronal death and reduce the capacity for a group of cells called microglia to eliminate the A-beta proteins, which finally leads to memory loss in Alzheimer's disease. So again, we confirmed impaired mitophagy is a culprit, not a bystander, because when we genetically and pharmacologically reinstall mitophagy, which leads to mitochondrial quality improvement, as well as survival of neurons and improved garbage elimination capacity by microglia, and finally contribute to memory maintenance in both C. elegans and 2 mouse models. Impaired mitophagy and reduced NAD not only shown in AD, but also has been shown in Parkinson's disease, ALS, and Huntington disease. Postdoc Sophia Lothrop, she asked a question like, is there a pivotal and shared pathway in those neurodegenerative disorders? To this end, we made a hypothesis to cell metabolism last year. We highlight the importance of the NAD mitophagy axis, the impairment of this pathway in different neurodegenerative disorders. Indeed, NAD reduction was low in Alzheimer's disease, in Parkinson's disease, in Huntington, and in ALS. So we highlight that this pathway is important, but not essential. We needed to have ApoE4 pathology and others to contribute to AD and for the others. We are very actively involved in translational studies. So I have been very fortunate to involve in a couple of premature aging clinical trials, as well as in neurodegenerative clinical trials, including Alzheimer's disease, Parkinson's disease, and ALS. Because we found that mitophagy induction contribute to memory maintenance and healthy longevity. So we are actively involved in using AI to prepare drug development in collaboration with a Germany and London-based company, Aladdin. My postdoc, Chen Longxie, he only used 8 months and developed this pipeline, including set up the library, modeling, biological validation, and finally, he identified 2 lead candidates from a library of 5,000 molecules. Those 2 compounds were able to induce mitophagy in both cells and worms, and also it can dramatically improve the memory in tau and A-beta C. elegans, and also in mice. So we are excited of the results and are going to wrap up. This is in collaboration with Aladdin, Imperial, and University of McCall. I want to give you a summary of my talk. So firstly, we show NAD depletion and mitochondrial dysfunction happens in different neurodegenerative premature aging disorders. NAD can regulate mitochondrial homeostasis, at least partially through mitophagy. When we giving the AD mice treated with NAD, we can improve the memory, which can contribute by elimination of tau and A beta. Due to the importance of mitophagy and autophagy, we highlight this can be the 10th hallmark of aging. And we are very excited on the clinical trial stuff of the NAD-based clinical trials. I want to take this opportunity to share with you because we know aging is so complicated, it can't be done by one people. So when I joined the University of Oslo 3 years ago, working with these 3 other co-founders, we initiate this Norwegian Center on Healthy Aging. So No Age Center. This is network now. We highlight cellular, individual, and societal level. We are actively doing meetings and conferences. So we welcome you to follow us and involved in our activities. And finally, I'd like to thank my team members, collaborators, funding, and conflict of interest. Thank you again, Morten and Alice, for this opportunity.
Yes, thank you, Evandro, for giving such a nice talk. We will need to stick with the schedule, even though I don't like breaks, but a lot of people need them. So I'll try to summarize the questions to the best of my abilities. And I'll start with the most upvoted one by Wilbert Vermeij. Other progeroid syndromes that do not show reduced NAD levels?
We have been working on ATM, XPA, cocaine syndrome, and almost all show reduced NAD, except XPC. We didn't find the phenotype.
Interesting. And so when does NAD start to decline significantly in aging? And what would be the expected target age group to start clinical trial?
Yeah, this is a very good question, because NAD even is very popular, but we do not have strong evidence when is a time point can dramatically reduce. We are lacking of the data from human population. However, from the C. elegans, we argue, at least from middle age, there's a partial reduction, and late age, there's a dramatic reduction. When to giving the NAD or to what target of population? Because the NAD precursor NR was in the market in the USA as a diet supplement from the early 2010s. So relatively it's safe, but whether it can treat any disease or not, I would be more stringent, try to waiting for the result from the clinical part before I want to give you any comments.
Excellent. Thank you, Evandro. There are more questions in the Slack channel, so I see that you are on it. It would be great if you can go ahead and answer those, because there is interest in this work. And